This gut bacteria discovery could change how we age. Did you know that some of the most powerful anti-aging tools in your body aren't in your DNA, your skincare, or your supplements, but in the trillions of bacteria living inside your gut? For decades, aging was treated as something we could only slow down from the outside. But today, scientists are discovering a radical new idea. What if we could turn gut bacteria into anti-aging factories, programming them to protect our cells, reduce inflammation, and extend healthy lifespan from within? In this video, we'll explore the breakthrough science behind this idea, why it could change how we think about aging forever, and how the future of longevity may begin in a place most of us have never thought to look: our own microbiome. Have you ever wondered why aging happens? Or if it's possible to slow down or even reverse some aspects of it? Most health advice focuses on diet, exercise, and sleep. But what if the key to healthier, longer life isn't just in your own cells, but in the trillions of tiny microbes living inside your gut? In today's video, we're exploring a groundbreaking discovery where scientists are literally turning gut bacteria into anti-aging factories that produce compounds linked to longer life. This research could reshape how medicine approaches aging, metabolism, and chronic disease. It's biology we've never seen before, and it might be a clue to living healthier for far longer. Background: the age-old question of aging. Aging is universal, a complex process that affects every part of the body over time. As we get older, our risk of chronic diseases like heart disease, diabetes, cancer, and neurodegeneration increases dramatically. Scientists have spent decades trying to uncover the biological mechanisms behind aging and to find ways to delay, slow, or prevent its worst consequences. While lifestyle factors such as diet, exercise, and sleep remain essential components of healthy aging, molecular research has uncovered internal processes behind cellular decline, DNA damage, metabolic dysfunction, and chronic inflammation that all contribute to aging. One promising area of study has been the role of the gut microbiome, the vast ecosystem of bacteria, viruses, and fungi in our digestive tract. Traditionally, research on aging focused on the body itself, cells, organs, hormones, and genes. But recent findings suggest that aging is not all intrinsic. These discoveries reveal that the microbes living in and on us influence how our bodies function far beyond digestion. They play roles in immune regulation, metabolism, inflammation, and even nervous system health. Now, scientists are discovering that these tiny organisms could be leveraged for anti-aging benefits in ways we never imagined. The new discovery, coaxing bacteria to produce anti-aging compounds. In a groundbreaking study led by researchers including Genelia's senior group leader Meng Wang, scientists found a novel way to influence gut bacteria so that they produce higher levels of a compound associated with extended lifespan. Here's how it works. The target: gut microbiota. Inside our digestive system resides a diverse microbial population known as the gut microbiota. These bacteria do a lot more than help break down food. They produce vitamins, train the immune system, regulate inflammation, and communicate with organs like the liver, brain, and heart. The compound cholinic acid. The researchers focused on a natural compound called cholinic acid. In earlier studies, cholinic acid had been shown to extend the lifespan of simple organisms like roundworms and fruit flies. However, the challenge was how to get this compound in significant quantities within an organism without introducing other risks. A low-dose antibiotic trick. Rather than injecting cholinic acid or trying to synthesize it in the lab, the scientists found that they could encourage gut bacteria to produce it themselves simply by exposing them to a very low dose of an antibiotic called cephalosporin. This doesn't eliminate the bacteria. Instead, it shifts their gene expression toward producing more cholinic acid. This is a radical shift in strategy. Rather than targeting the host, the animal or human, they targeted the microbiota, essentially programming the gut bacteria to act as chemical factories for beneficial compounds. Understanding cholinic acid? Before we go further, it's important to understand why cholinic acid matters. Cholinic acid is a type of oxalopolysaccharide, a complex sugar produced by certain bacterial strains. In simple organisms like worms, Caenorhabditis elegans, increased levels of cholinic acid have been linked to robust longevity effects, including increased stress resistance and enhanced lifespan. Until now, however, it was unclear whether similar benefits could be translated into mammals such as mice and, eventually, humans. The breakthrough comes from triggering the gut microbes to make more of this molecule. Since gut bacteria already play a huge role in metabolic signaling and immune interactions, enhancing their production of beneficial compounds could directly influence how the host ages. This isn't merely supplementation, it's about leveraging the microbiome as an internal drug factory. In this study, animals exposed to low doses of cephalosporin showed measurable increases in cholinic acid production by their gut microbes. And the effects were not small, they influenced key aspects of aging physiology. Roundworm results, longer lives, and healthier metabolism. The researchers first tested their approach in Caenorhabditis elegans, a tiny roundworm widely used in aging research due to its short lifespan and well-mapped genetics. When these worms were treated with low-dose cephalosporin, their gut bacteria began producing much more cholinic acid. The results were striking. The worms lived significantly longer than those not exposed to the treatment. This provided initial proof that the strategy had promise and helped establish a direct link between bacterial metabolic changes and host lifespan extension. It also reinforced that gut microbiota are not just passive passengers. They can be active contributors to longevity pathways. Mice experiments, metabolic health improvement. After the success in roundworms, the scientists tested the strategy in mice to see whether similar benefits might appear in mammals. In mice, low doses of cephalosporin did not get absorbed into the bloodstream, meaning the antibiotic remained in the gut where it could influence bacterial gene expression without exposing the rest of the body to potential side effects. This is an important safety consideration often overlooked in drug design. Here's what they observed. Metabolic shifts. Male mice had higher levels of good HDL cholesterol, which is linked to lower cardiovascular risk. Bad cholesterol LDL levels decreased, supporting healthier lipid profiles. Female mice exhibited lower insulin levels, a key marker for improved metabolic regulation and reduced diabetes risk. These changes suggest the gut-mediated production of cholinic acid influenced systemic metabolism, even though the antibiotic itself never entered the bloodstream. This points to a fascinating therapeutic strategy, targeting bacterial metabolism as a way to modify host physiology. Instead of directing drugs at human cells, we may one day direct them at the microbiome to produce health-supporting compounds internally. Why this approach avoids toxic side effects? One of the biggest challenges in drug development is toxicity, the unwanted effects that occur when a drug is absorbed by the body and interacts with various tissues. Many promising anti-aging compounds have never made it to human use precisely because they cause collateral damage. In this study, cephalosporin's lack of absorption into the bloodstream made it uniquely suitable for microbiome targeting. It stayed in the gut, shaped bacterial behavior, and stimulated the production of beneficial compounds without harming other organs. This is a powerful concept. The microbiome itself becomes the therapeutic agent, producing health-promoting molecules where they are naturally made in the gut, without systemic drug exposure. If translated to humans, this could dramatically reduce side effects while maximizing benefit, a major step forward compared to many current medications that are distributed throughout the body after ingestion. Implications for human aging and longevity. So what does this mean for human health? We're still years away from applying these findings directly to people, but the scientific implications are profound. A new paradigm in medicine. Instead of focusing solely on human cells or organs, medical research could increasingly leverage the body's microbiome as an internal factory at crafting beneficial compounds that support health and longevity. Broad health applications. Because the gut microbiome interacts with so many systems, immune, neurological, metabolic, and hormonal, this strategy could have ripple effects beyond aging alone. Microbe-directed therapeutics. This research supports a future where drugs are designed not to act directly on human tissues, but to co-opt beneficial microbial pathways. That means fewer side effects, more targeted outcomes, and therapies that work with the body's natural ecosystems. While translating animal research into human treatments is always complex and requires extensive clinical testing, this discovery opens a door to entirely new therapeutic approaches that could revolutionize how we think about aging and disease prevention. The idea that our gut bacteria could be turned into anti-aging factories may sound like science fiction, but this study brings it closer to reality. What started as a curiosity in worms has now shown meaningful health effects in mammals. As researchers continue to unravel how the microbiome influences aging, metabolism, and immunity, we may find that our inner microbial communities are among the most important allies in the quest for longer, healthier lives. If you enjoyed this dive into cutting-edge longevity science, hit like, subscribe, and tell us in the comments what part of this discovery surprised you most. Biology may have just given us a new way to think about aging, not as something inevitable, but something we might influence from the inside out.